afternoon, everybody. This is Dr. Tuğba Selimoğlu. I am the head of the Bioengineering department and the Vice Dean for the Faculty of Engineering and Natural Sciences. Today, I'll be talking about bioengineering in general and some of the major areas of bioengineering. Before I move on to my presentation, I would like to share a nice quote that I like. It's from Oliver Holmes. It says, "The mind, once expanded to the dimensions of larger ideas, never returns to its original size." So before understanding what bioengineering is, we have to understand what an engineer is. An engineer is actually a problem solver, independent of his or her specialization. So as engineers, we would like you to solve problems and be a problem solver, actually. So what do we do? What do engineers do? Engineers use math and science knowledge and to find innovative ways to solve problems and design new products and technology. What's our mission? Our mission is to connect the discoveries made by scientists at the lab level and transform them into products made at the factory level and scale them up as a profitable end product for society. So what's the engineering process? Engineers solve problems, but so do mathematicians. Engineers analyze, and statisticians analyze as well. So what is the distinguishing characteristic of an engineer? Well, the distinguishing characteristic which separates an engineer from other fields is that it is concerned with the design of systems. This design involves a considerable amount of creativity, and it's kind of an art that one learns through experience. So let's move on to bioengineering. What is bioengineering? We have a really nice and long definition, a formal definition. It says it involves the design, construction, and operation of unit processes that involve biological organisms or molecules, and is involved in the biotechnology, pharmaceutical, food, environmental, and waste treatment industries. Well, bioengineering is a diverse area, and each bioengineer can only learn and know a tiny fraction of the entire field. Let's have a shorter definition. Bioengineering applies engineering methods and techniques to problems in biology and medicine. We make use of several other fields, of course. It's a diverse area. So the most important one is mathematics, actually, which I haven't written here. And biology and medicine, material science, chemistry, physics, nuclear engineering, computer engineering. These are all the areas we make use of, and some more. Here you can see a figure where the intersection of other areas with bioengineering, actually. For instance, if we want to design biological systems and prosthetics, we can make use of mechanical engineering. And if we want to do computational biology, computational and molecular and cellular biology, we could make use of biology and neurobiology. And if we want to go into biomimetics, we could use material science and engineering. So let's move on to some of the major areas of bioengineering here. I'll be talking about bioprocess engineering. What is bioprocess engineering? It is the area that deals with the design and development of equipment and processes for the manufacturing of products such as agriculture, food, feed, pharmaceuticals, and treatment of wastewater. These engineers develop concept technologies in the bioprocess space. So if you want to produce a product from biological material, we are a bioprocess engineer. What is a biological material? It could be yeast, fungi, bacteria, algae, viruses, mammalian cells, or any type of biological single-cell process. So here I have a workflow of a bioprocess, is the production of polyhydroxyalkanate using cyanobacteria as a host. You can see the upstream processing, the fermentation process, and downstream processing here. I'll be explaining these processes a little bit further. What is an upstream process? It is the first step of the bioprocess engineering, in which biomolecules are grown actually, usually by bacterial or mammalian cell lines in bioreactors, which I'll be explaining a little bit later. So when these biomolecules reach the desired density for batch and fed batch cultures for bioreactors, they are harvested. So this is the step before the bioreactor. And after the bioreactor, we have the downstream processes. So actually, upstream refers to the material inputs needed for production. So we have the material input, the production, and the separation. Upstream process, fermentation, and downstream process. These are all similar terms. So we talk about fermentation process. We sometimes use fermenter, sometimes bioreactor. So what is a fermenter or a bioreactor? Well, actually they are the same thing, and they are the central part in the bioprocess because they provide the link between the raw materials and the final products, and we conduct fermentation in those. It is a vessel in which a fermentation process is carried out. Fermenter is used, but bioreactor is used in a wider sense, because we could say that a lake is a bioreactor or a transgenic plant is also a bioreactor. So we choose to use bioreactor as the general term. So what is a bioreactor? It's an apparatus, actually. It's a jacketed cylindrical vessel for growing organisms such as bacteria, viruses, or yeast. And we use them in the production of pharmaceuticals, antibodies, or vaccines, or for the bioconversion of organic wastes. And we need optimum conditions for a bioreactor. What is the optimum condition? The flow rates, the temperature, the pH, dissolved oxygen level, agitation speed, the microorganisms or cells that are needed to reproduce at a rapid rate. Okay, so we have covered upstream processes and then the fermentation, and downstream processes comes later. What is a downstream process? It refers to the recovery and the purification of biosynthetic products, usually pharmaceuticals from natural sources such as animal or plant tissue, or fermentation broth, including the recycling of salvageable components and the proper treatment and disposal of waste. So there are some basic operations in downstream processes we might like to talk about. So if you want to remove biomass from the fermentation broth, we could use centrifugation or filtration or sedimentation. If we want to look into the concentration of the raw product, we could use extraction or adsorption or distillation. And for the final purification of the final product, we could use chromatography, decolorization, or crystallization. There are different operations in downstream processes that we could use. And these are not limited to the ones that I'm showing here. So what's the difference between upstream process? It's the upstream process is the bioprocessing phase in which cell lines are generated. The downstream processing is the phase that deals with extraction or separation of desired products from the biomass. So let's move on to another area of bioengineering. It's computational bioengineering. In this area, we use computational tools to analyze large biological data sets, such as in genomics or proteomics. And we can use computational models to predict structure and behavior of large biological molecules. And we could also design new drugs. This area also includes bioinformatics and also modeling of biomolecular structure, dynamics, and interactions. We could do protein and peptide design, and we could model cellular signaling pathways, and we could also do drug discovery. So here is a figure for the application of bioinformatics tools. So if we are in the area of bioinformatics, we could do molecular modeling, molecular interactions, phylogenetic analysis, protein sequence analysis, drug design, molecular dynamic simulations, and DNA sequence analysis. And for these, we could use some bioinformatics tools. Databases are really important in bioinformatics, mostly NCBI, PDB, SwissProt. Those are the databases that are mostly used. And for sequence alignment tools, we could use ClustalW, FASTA, hidden Markov models. And for 3D structure alignments, we could use PRISM, CATH, or SCOP. Let's move on to another area of tissue engineering. What is tissue engineering? In tissue engineering, we use engineering design principles to regenerate natural tissues and create new tissues using biological cells and three-dimensional scaffolds of biomaterials. What are the candidates for tissue engineering? We usually use skin, cartilage, heart, and bone as tissues for tissue engineering. How would we do tissue engineering? Well, if we have complex wounds, such as burns, we could produce skin substitutes. So we could use them in skin graft surgeries, and this is kind of tissue engineering. So what do? We actually get a biopsy from the patient, and we do cell isolation and cell culture and cell proliferation, and we grow this tissue on a scaffold in a bioreactor with chemical and mechanical stimulus, and we implant it back to the patient. Let's move on to another area. This is also a big area and an important area for bioengineering. It's called drug delivery. It's the design of materials and systems to achieve controlled release of drugs in physiological systems. When we talk about drug delivery, we're actually talking about approaches, formulations, technologies, and systems for transporting a pharmaceutical compound, which is a drug actually, in the body based on nanoparticles, as it's needed to safely achieve its desired therapeutic effect. Here we can see biologists, clinicians, material scientists, and engineers in the design level of a drug. And then we can see the synthesis and production of a drug in a nanoscale, a microscale, or a macroscale. And then we can see the treatment of conventional release profiles or on-demand release profiles, because controlled release is important for a drug and the treatment. So here, you can see some drug delivery systems that are used, which are nanoparticles, micelles, dendrimers, hydrogels, liposomes, solid lipid nanoparticles, gold nanoparticles, magnetic nanoparticles, carbon nanotubes. So we have a lot of options for drug delivery systems. There's one more area that I'm going to be talking about today. It's genetic engineering, and it's the process of using recombinant DNA technology to alter the genetic makeup of an organism. One of the earliest uses of genetic engineering was to mass produce human insulin bacteria in bacteria. Now we can produce human growth hormones, human albumin, vaccines, and many other drugs using this technology. Here we can see gene transfer method in plants. So first, what we do is we insert the gene into a plasmid using a restriction enzyme and DNA ligase. And then we introduce it to plant cells in culture. And finally, you can see the regeneration of the plant and plant with nitrate. And if we want to show it in animals, it's obviously different. We have an organism to be cloned, and we take the differentiated cells from that organism. In this case, it's a cow or ewe. We have an unfertilized egg cell, which the nucleus, we remove the nucleus from this egg cell, and we take the egg cell with no nucleus and develop differentiated cells from the donor, and we fuse them together. And this fusion begins to develop as an embryo, and it's implanted into an ewe, and a clone is born. So today, this is all I have for you. I hope that you follow us, our official account of Bioengineering Department on Twitter from this account. And we also have a Bioengineering Club that you guys might like to follow. And I gave the address here too. So have a nice day and hope to see you soon. Bye.